Dr. Abba Zubair. Dr. Zubair currently is the Medical Director of Transfusion Medicine and Stem Cell Therapy at Mayo Clinic, Florida. Dr. Zubair is also the Associate Dean of the Mayo Clinic School of Health Sciences. Dr. Zubair just published the results of a spaceflight experiment in June, and today he will discuss the expansion of human mesenchymal stem cells for the benefit of humankind on Earth and beyond. Dr. Zubair.
Thank you so much for the nice introduction. It's really a pleasure to be given this opportunity to talk to this audience. So as was introduced, I'm going to talk about projects that went up to the International Space Station. With the project's primary objective is to evaluate the feasibility of expanding human mesenchymal stem cell. These are primary cultures, not cell line, but freshly derived human mesenchymal stem cell expanded at the International Space Station with the intention of bringing them back to us for human application. We have several clinical trials going on that involve mesenchymal stem cell, and we believe that would form a platform of future human application of space-grown cells. So why taking stem cell to space? And why do we utilize microgravity? I think the short answer lies on this principle. Obviously, we're talking about growth or expansion. So at the heart of it is cell division. And when cell stem cell in our body divide, they undergo what we call asymmetric self-renewal. Basically, one stem cell would divide into two daughter cells. One daughter cell would retain all the characteristics of the parent and stay as stem cell, while the other continue to proliferate and give and differentiate and give rise to matured cells. In this process, stem cells are able to maintain their number constantly. So it's much harder to really expand stem cell, even though we found tricks in the lab where we can expand them. But inherently, they are designed to keep their numbers so that at any given time, the proportion of stem cell is about the same, which is a big challenge if you are planning to expand and grow cells for a number of patients, not just one patient, but giving it to multiple patients. So you need to grow it, and that's the challenge. If you normally cells that are not stem cell undergo symmetric differentiation. So basically the cells divide and the two daughter cells do not have the characteristic of the stem cell. And these daughter cells further differentiate to give different types of cells. Obviously, with induced pluripotent stem cell technology, you can reprogram the daughter cells to be back to having stem cell characteristics. So the main question is whether we can be able to induce symmetrical cell expansion or stem cell expansion under microgravity condition. We know gravity play a role in virtually all our physiology. I tell my students that if human race evolved on Jupiter or Saturn, we wouldn't look like the way we look now. We would be a lot more like a lizard or something that is more flat, because the gravity there is much, much more than on Earth. So we know that gravity play a role in cell division, in chromosomal segregation. We also know gravity impact on gene expression as well. So the question is, if you take it out, would you be able to reprogram these cells safely to expand without, making them unsafe. So we chose mesenchymal stromal cells. Some people call it stromal cells. Some call them stem cells. So MSCs are actually maybe the most common or second most common cells that are used clinically in regenerative medicine. They can be derived from bone marrow or cord blood, cord tissue, or adipose site, so from fat. So the access is quite reasonable. They are defined by some markers, CD73, CD90, and 105, or absence of some markers, which are mainly differentiating them from hematopoietic cells. All these markers are not unique to the MSCs, but a combination of them kind of define the MSC. The International Society for Cellular Therapy also add to this definition that MSCs should have the potential to differentiate into bone, cartilage, or fat. So osteocyte, chondrocyte, and adipocyte is one of the key characteristics of MSCs. So I started by mentioning that obviously stem cells are used clinically, and so they are highly needed. Patients can donate their own stem cell, but not every patient are able to do that. Some of the patients, for example, that have leukemia or lymphoma, their marrow may not be adequate for even donating the cells. The stem cells themselves are damaged. So there is definitely need to have a donor or a healthy donor to donate. But there aren't many healthy donors that are available to donate. And then even if they do, it's harder to expand the cells to treat more than one donor. And we know based on, let's say, the bone marrow transplant activities that we do, we need about 200 to 500 million stem cells to achieve therapeutic efficacy. Again, in the setting of, let's say, leukemia, lymphoma. Of course, some application where you are putting the cells locally, you may even need just 5 million or 10 million or less. But regardless, you need a lot of cells to treat a lot of people, especially if you have one donor donating to multiple recipients. So we don't have effective method to really expand the cells. We do have some method, but it takes time. At a minimum, you need about a month to grow a reasonable number of cells to treat a few patients. And even if you need up to like 200, 500, you may need a lot more, or you need a bigger facility, a lot more resources. So scaling up is always a challenge, and you can see that the space are limited. You need more incubators. We are using multi-layer flask. All of that, and there's even automated bioreactors, all of that is quite pricey and difficult and takes time. So we asked the question, can stem cell expand in microgravity safely? What is the feasibility of doing that, especially if you want to generate clinical grade cells? So yeah, so we want to evaluate the safety and efficacy of using these cells for clinical application. Obviously, the journey to International Space Station was quite exciting. It takes time to get on the bus. What I mean by the bus, the only rocket at the time when we went is SpaceX that could take research payload. So you need to be on the queue, and for us, it takes almost 2 years to get on the rocket. This is Peggy Whitson. I tell people she's the most expensive lab technologist I've ever had. obviously being the astronaut that worked on our project. So it's really a pleasure and a great opportunity to have an astronaut work on our project. And luckily, she also have a biology background, so that really helps. So what you are seeing is the container, the cassette that we use, BioCell, in collaboration with our implementation partner, which is the BioCell from University of Colorado. And that's how we were able to transport the cells in a PHAB and then to the International Space Station. It's quite challenging. We had initially thought we can do a lot more at the station, evaluating the cells, but reality set in, so we had to scale down a lot of things. In part, you got an astronaut who may have up to 20 projects working on yours, so you are just one of the many who is not really a basic researcher, so you have to really scale down. And then the setup also takes quite some time, like the use of microscope and stuff like that, or media exchange. We had to even do away with that in space. But anyway, we took 3 different types of cells: hematopoietic stem cell, leukemia cell line, and then the mesenchymal stem cell. So this presentation really focused on the MSCs, but we also look at other cells. We were able to get real-time imaging as the cells grow, and then we took the same pictures of the control cells in our lab. So you can obviously see it wasn't that clear, but at least for the leukemia cell line, the ones that are growing at the International Space Stations form clusters, while the ones that are growing in our lab didn't. And you can see also, it looks like the MSC are kind of more enlarged compared to the ones that are in our lab. So, but it gave us some clue. But that's all we could do at the International Space Station. Beyond imaging, we just preserve the cells in multiple ways, some in RNA protect, some because we want to further grow the cells when they come back to our lab. So we use cryoprotectant DMSO, dimethyl sulfoxide, and some we just fix the cells in formalin. So we had multiple ways of preserving the cells so that we can do further analysis when they return. So they return via this return capsule. Actually, this is the exact capsule containing our product. It landed somewhere in the Pacific Ocean. It get fish out, I mean, obviously using GPS. And then actually our samples were mailed by FedEx. So from, International Space Station down to us, and then the FedEx pick it up and deliver the cells to us here in Mayo Clinic, Florida. So it's quite remarkable that you can get your cells back by FedEx and safely. So when the cells get back to our hands, there's so much excitement, and the people were asking, what is the result? I tell people, well, wait a minute, that's actually the real experiment start when we get the cells, because we couldn't do much at the International Space Station. So, and we were very careful not to throw out all our cells all at the same time, because these are precious samples, so we were very careful. But we did cell characterization, looking at cell count, viability. We did cell cycle analysis, gene expression profiling, growth factors, and then also look at differentiation. I may not be able to present all the data on this, but it was quite remarkable, and more to come, and we're still doing the analysis. The very important mission for the objective for this mission is really safety of the cells. So we wanted to see whether there's any radiation damage, so like DNA damage of the cells. Also, we wanted to see if they transform. So, and then we want to see whether they retain their regenerative capacity and induce tissue repair. So that is still ongoing, and I wouldn't be able to touch on the efficacy beyond the immunosuppressive activities. So quickly, these are the characteristics of the cells, you know, before flight. You can see the cell identity that expressing those three markers is quite high, 89% viability, 96. And then these are the cells that we started with. Obviously, they are sterile. The gram stain and cultures were negative. We culture the cells for 1 week and for 2 weeks. So the data on the 1 week cells, you can see the identity is comparable between ground, which is a control, and the cells that were grown at ISS. You also can see the viability is reasonably comparable, even though the viability is a little bit lower for the ISS compared to the one in our lab. We are suspicious about maybe this is just due to the way this has been cryopreserved. We couldn't use control rate freezing, but I mean, obviously it has traveled this far, so maybe the viability is a little bit less. The cell growth is also, actually we see a little bit more cells on average, but overall is within the error of margin. We think the growth rate is comparable. And the same for the 2 weeks culture as well. The viability is a little bit worse, especially the ones at ISS. But again, we think that could be also related with the cryopreservation method. All of this is sterile, and that's very remarkable. We're worried of microbial contamination during processing or transportation, but all look good. We also tested their ability to proliferate, so we saw the cells and further culture them. The figure A is looking at real-time imaging using IncuCyte as the cells proliferate, and then on the figure V is showing the population doubling time. The summary of it is that we didn't see any significant difference between control versus the cells that were expanded at ISS. So no impact really on proliferation as far as we can tell. We also look at, of course, we are talking about cell expansion, so we look at cell cycles and look for expression of cell cycle checkpoints, inhibitors. So CDKN2 is a cyclin-dependent kinase 2A, which inhibit transition from G1 to S phase. And then we also look at E2F1, which is interact with RB retinoblastoma protein, which is a powerful cell cycle regulator. And then the polo-like kinase 1, which is serine threonine kinase, which inhibit transition from G2 to M phase of cell cycle. As you can see, at 1 week culture, there is really no significant difference. So also in 2 weeks culture, other than the PLK1 is significantly decreased compared to the ground control. We really think more studies needs to be done, but clearly it looks like microgravity might be inhibiting the MSC expansion or progression in the cell cycle. But I think more studies need to be done. And then we look at differentiation, both using using stain for osteogenic differentiation and the oil red for adipogenic differentiation. The figure A on the top is just showing normal cell grown in regular media and then the differentiation media. We also look at genes that or transcription factors that are known to be associated with osteogenic differentiation, which is the first two, the CLK1A and the RANKS X2, and then the CRAD B is for adipogenic differentiation. And the summary of it is that we haven't seen any difference between control and space-grown MSC in their ability to differentiate. We also look at condition media for cytokine release, looking at both ISS-grown and ground control. And the interesting thing is that we look at 65 different cytokines, and we were only able to detect significant difference in two. There's SCD40 ligand, so which is significantly decreased, and then the platelet-derived growth factor is significantly increased after 1 week of culture, but we see most cytokines being affected after 2 weeks of culture. For example, the IL-10 is significantly decreased. The MCP3 also significantly decreased. So as the SDF, which is a powerful chemokine for attracting cells and impacting on cell migration, that also is decreased. This was previously reported, so it's interesting to see our studies also confirm previous report. And then also the IP10 is increased. I think, again, with this one, the conclusion you can only draw on this is that, yeah, cytokine and growth factor expression is impacted by the absence of gravity. But more studies would be needed to really comprehend the downstream effect of these changes. We also look at potency, assay. We wanted to see if the MSC are more immunosuppressant than when they were grown at under microgravity condition. MSCs are known to be immunomodulatory, and actually it's one of the primary reasons that they are being used. So they are used in treating rejection of organs, also used in as immunosuppressant to control graft versus host disease after bone marrow transplant. So we wanted to see whether that properties of theirs is maintained or enhanced or decreased. And the short answer is the MSCs that are expanded in space are much more immunosuppressive than the control, both at 1 week and 2 weeks. Finally, we come to safety. We are worried about DNA damage. So we use this marker that is this transcription factor as part of the histone family of transcription factors that get exposed if the DNA is damaged. So we stain the cells, and then we also expose the cells with etoposide, which has DNA damaging properties as our positive control. We also use DAPI to stain the nucleus, so that we wanted to show that the damage, or the DNA damage, is in the nucleus. So basically, you can see when the drug is used, you can see a lot of DNA damage, and without the drug, there is no damage. And we haven't seen difference between gram control and space for all the weeks of culture. We also did chromosomal studies and karyotyping. This is a gene banding, and clearly we didn't see any chromosomal abnormalities when we compare the ground control and the ISS. And then finally, we said, well, what about tumor formation? So here this is a positive control. We're using a fibroblastic sarcoma cell line. You can see the colony being formed, and this is what we are looking for. So, and then this is a negative control of just the cells, different from the ones that are used in the control group. So you can see the ground in 1 week, and then the microgravity ISs in 1 week, and then in 2 weeks. Both there is no evidence of tumor formation like what we have seen in the positive control. So the summary of our finding is that so we asked fundamental question: do MSC survive and grow faster in space under microgravity condition? And the short answer, yes, MSC grow in space under microgravity condition, and their growth is comparable to the one on the ground control. So we didn't see that further expansion where we can grow more cells. But these are the MSCs. We've seen some expansion on the HSC, and there wouldn't be time for me to go into that data, more to come down the road. And then in terms of feasibility, growing MSC in a sterile and safe way at the International Space Station, clearly we can see that we can do that. MSC can be cultured without any microbial contamination. We show that they haven't been transformed to form tumors. They have normal chromosome. The karyotype is normal. No DNA damage seen. We also have seen that there is an effect of the microgravity on the MSC capacity to secrete some cytokine and growth factors, and that the MSC actually that they are more immunosuppressive compared to the ground control. So those are the main findings. Obviously, we have a lot more questions, so it's really nice to be given this opportunity, and thank you so much. I will stop now. Thank you.